you so much for your introduction. I appreciate the invitation of NCARB, very exciting meeting. Let me start. So today we're going to be talking about COVID-19, particularly the molecular testing aspects. Before I start talking about the testing options, I'm going to introduce the background on SARS-CoV-2 virus. So this virus belongs to beta coronavirus. It's named corona due to the structure of the S protein around the virus. It looks like a crown. And the first version of this virus is SARS-CoV, which was, discovered, in the early 2005. And there is another version of corona that is the MERS coronavirus that is slightly different in genetic makeup. I don't think I need to go through the details on how the virus infects. We know that the infection aspect happens due to the S protein, and I'm going to go into the next level, next slide. So the S protein, S protein in yellow is the protein that is involved in invasion. Then we have a first open reading frame right here, open reading frame 1A and 1B.
We can't see your slides.
Oh, you cannot see the slides. Okay, let me see if I can share. Are you able to see the screen right now?
Yes, perfect.
Thank you.
Yeah,
thank you. Thank you.
Yes.
Perfect. So I was saying earlier that the coronavirus genome has about 30,000 base pair. For being a virus, it's a substantially large genome compared to other viruses that we have seen. Now, the virus is divided into three main domains. First one is the, I'm going to start with the yellow one, which is the S protein. This is the protein that the virus uses to enter the cell, so it's for invasion. And then you have the open reading A1 and B1, and 1A, 1B. Those are this is the open reading frame that encodes for a replicase enzyme. So unlike other viruses, this virus rely on open reading frame 1A, 1B to do the rest of the RNA replication and translation. So first you have the first open reading frame translated into a replicase enzyme that will take care of the rest of the genome. Very interesting way of infection. Now the last part, the 3' encodes for all the enzymes required for packaging and release. And that's the genome of coronavirus SARS-CoV-2. Most of the probes that are designed for RT-PCR, we're going to go into that later, they are designed against this open reading frame and then S and N. And sometimes you find a target against E protein right here. So those are the proteins that are made by the virus. We have 14 open reading frame, 27 proteins. When it comes to the diagnosis options available for COVID-19, first option, the most commonly used, is screening by symptoms. So you know that a lot of clinics and hospitals are looking for symptoms like cough, fever, etc., a short breath or respiratory symptoms. Then we have a more drastic diagnosis, which is the CT scan. On the molecular base, you have the nucleic acid detection, which is RT-PCR, or you have the rapid isothermal amplification. So these two tests, both are part of the nucleic acid detection approach. Then you have the antibody. So the antibody, you have the lateral flow test, which is the rapid test, and you have the conventional test, which is the ELISA test. So when you look at this slide, for example, this is just looking at the common symptoms and how often they have been registered in patients for fever and cough. So fever, for example, 88.7% of patients who have coronavirus were recorded to have fever. So it's a pretty good way of doing the initial screening. Obviously, you have to add other molecular tests to it, but it's a good starting point. Cough counts for 67.8%. Back to the molecular testing, COVID-19 RNA test, a way of detecting RNA, because coronavirus is an RNA, single RNA genome. So you have two tests. You have the rapid test and you have the conventional test. The conventional test is a qPCR. We call it Q for quantitative PCR. And it relies on fluorescence detection. And I'm going to go into that in a little bit more details later. Then it takes about 3 to 4 hours. The extraction, the preparation, extraction, the testing, the result, interpretation. And typically an RT-PCR should be in the 90s, at least 90% sensitivity. When it comes to rapid tests, it doesn't use PCR reaction. PCR is literally, PCR equipment is a machine that will heat up the temperature up to 96 degrees to separate the DNA, and then cool down so that the primers that amplify the DNA will align to the genome and amplify, and then it will increase again to allow the amplification. So it's a change of temperature, continuous change of temperature for many cycles, up to 40 cycles. And that's the PCR. The rapid test works completely differently. It uses a stagnant, constant temperature, and it uses enzyme that will open the DNA, amplify through them, then the amplified DNA will drop, then it will continue the cycle. It will open, amplify, and that happens at room sometimes at 40 degrees, but the chemistry and enzymes used vary from test to test. In this case, it happens at 65 degrees. So 65 degrees is the optimum temperature for isothermal amplification. The Abbott test uses a similar concept. It uses an isothermal amplification, and that's why they claim the test takes only 15 minutes. So in each test, you have two stages. So you have the amplification and you have the detection, right? So in this case, the detection in the rapid test can be either colorimetric or it can be fluorescence-based. And for the conventional RT-PCR is fluorescence-based. Now, we have seen from media and from certain hospitals that the sensitivity of the test, of the rapid test, was not that great. In the media, they reported approximately 30% sensitivity. I don't have data, and I haven't seen data personally, so I can't make an argument or an opinion there. But typically, most rapid tests tend to be less sensitive. It's just an inherited limitation that you have to deal with when you are doing the testing. If you want the test that is highly accurate, and it will take more time, it will take more equipment involved, and that's why you're trading off sometimes, speed with sensitivity. Now, going into the isothermal amplification, this is just a diagram that shows you how the primers will recognize the RNA. It will start amplifying through the RNA, and then those primers will have a fluorescence, a marker that will fluoresce. As you have more amplification, you will have more binding, DNA binding, and therefore more fluorescence. To illustrate this in a little bit more details, if you look at these beacon molecular beacon strategy, so you have a fluorescent dye in red and you have a quencher in purple. When these two are very close to each other, there is no signal. But then when they are separate, then the fluorescence will come. And that happens when there are a lot of DNA amplified. The more DNA amplified, the more these dye will bind, and then the quencher will get separated, and then you have more signal. And so that's the principle of fluorescence detection using isothermal amplification. When it comes to qPCR, it's slightly differently. You have a DNA. The concept of fluorescence is the same. So you have a fluorophore and the quencher. And then the DNA, the enzyme will come and amplify, and then you have a change of temperature, then this DNA will separate, and then you have a new amplification, etc. So when you look at this figure at the bottom, that basically illustrates the amplification in each cycle. So you have a cycle that starts from 0 all the way to 40. The earliest you have the amplification, like in red here, displayed in red, the more RNA you have in the sample. So that's why it can be quantitative. And so if you have very little RNA, then you tend to detect the amplification later, which would be, in this case, a purple signal. And so in the y-axis is how much RNA or DNA, in this case, RNA translated into cDNA and then DNA. So this in y-axis is how much DNA you are detecting, and on the x-axis, when you are detecting that DNA. If you guys have any questions, please feel free to ask me either during the presentation or towards the end. So when you look at the genetic makeup of the virus, almost a few weeks ago, the rate of mutation was 229 that were recorded, 229 mutations. Now I'm sure it's in probably the 400 or 300 mutations so far. So I haven't looked at the last number, but the last time I saw it was 229. What that means in terms of diagnosis, if those mutations land in the place where the primers are designed, the primers may not work. Depends what position that the mutations occur, and it depends what strain of a COVID-19 you're testing. Because when you're testing different patients, you may be testing slightly different version of COVID-19. So there was a study showing certain strain where amplification did not happen because of mutation. Typically, genes that are targeted for amplification using qPCR are open reading frame 1B. So you have a probe here on one in S and one in N. Those in our lab, those are the ones that we use. Other labs have used gene E for envelope and then RDRP. So those are, and we use MS2 phage genetic material as a control for procedures. When you look at the outcome coming from real-time PCR, there is a lot of variability of results. We have seen papers that have shown very low false negative rate, and we have seen papers that have shown higher false negative rates. And really, this is the hardest thing to deal with when you are dealing with a body of literature that is spamming from an accuracy of 70% all the way to 96 or 95%. So what study to believe? This is the most common question we get. It's what is the sensitivity and specificity of the test? And this is really the hardest question to answer, and I'll tell you why. So looking at this study, for example, so you have 167 patients. They looked at the correlation between RT-PCR and CT scanning the chest. And they have seen that 155 up to 167 correlated perfectly. 5 patients were negative with RT-PCR, positive with CT, and 7 patients were positive in RT-PCR, negative in CT. So it shows, according to this, approximately a sensitivity of 68 in nasopharyngeal and 72 in throat swab specimen. If we look at a second study, and you look at, in this case, they took the CT scan, they took RT-PCR positive samples, and then they said, how many of them are CT scan positive? And so 97 were positive. When we look at all the RT-PCR tests, with confidence, I can say that the false positive rate is very low across all of them. The problem is the false negative rate, is when you think a sample is negative when it's not. So what causes the false negative rate to be high? And in this case, there are a few source of errors. One of them is the stage of infection. So if you get a patient that is too early, where there's not enough RNA in the system to detect, then you may get a false negative when the patient is just starting to pick up the virus infection. Another limitation could be the sample, how you collect the sample, and how do you take care of that sample, what is the transportation system, how long it stays before it gets tested, etc. As far as collection method, nasopharyngeal, although is considered to be the gold standard, it's sometimes hard to do. Sometimes you don't get the collection, the quality collection that you need. So you have to be aware of that collection being a source of negative, false negative results. And obviously the technology, you have to look at what technology you're using. If it's a PCR, qPCR, what probes are being used for that test. You have to have at least 3 probes, because one of the selection criteria that we do, or scoring criteria, if only 1 probe works and 2 don't work, then that test will be considered inconclusive. We have to have at least 2 probes that works, that gives you a positive out of 3 in order to consider that a positive test. So this is a very interesting study. You can look at the literature here. Maybe you can go and verify this. But what they looked at 61 publications, looking for publications that have quality data, so they can really determine what is the true accuracy of RT-PCR. 54 out of 61 were rejected based on inconsistency or lacking information or details that will really allow you to judge a paper or allow you to fully evaluate that paper. And that is just an inherited problem that we have seen with the COVID-19 time, because everybody is rushing to publish whatever data they have. Sometimes they have collected samples in the hospital, not using any scientific criteria, and they wanted to publish that. And you end up with a lot of publication all across the board. You don't know which one is true. So you have ones that show you 60% correlation or true negative. One shows you 70, one shows you 90. And so in this review, what they do, they looked at 61 available publication, rejected 54 based on quality data. They end up with 5. And if you look at these 5 research papers, 3 of them did not report the type of qPCR that has been used. And that's important because you want to know what primers have been used, and are they 1 or 2 or 3 type of genes that have been tested, and the specimen was not reported. Okay, going next, what specimen to collect? For now, we have seen labs starting with oropharyngeal and nasopharyngeal. Now we have starting to see saliva and sputum. This study just shows you that saliva is as good as nasopharyngeal. We have, in our lab, we have started to receive saliva sample, and we are in the process of evaluating that and comparing that to nasopharyngeal. Now, with this, I have ended my first part of presentation on the RT-PCR. I'm going to move quickly into the IgG and IgM. Similar to what I said on the RT-PCR, the IgG and IgM, the rapid test is what you see here. This is a cassette where you can take a blood draw and insert it in the S location. In this area, you have the antigen. So the concept is you have to have an antigen that is a peptide, a protein coming from a virus. Then you have your serum potentially having an antibody. The antibody and antigen will bind in this very first entry of these capillary or this lateral flow. And then because it has some pads, the pads will absorb the material, and the material will start moving through the membrane till it hits the conjugated antibody for IgM or IgG located here. When the antigen antibody find the conjugate antibody, it forms a complex of antigen antibody, conjugated antibody, and that leads to a marker being displayed there in red color. And so this test is colloidal gold test for a gold color, or red color that display in this test. This test has been widely distributed in the United States and all over the world. There are a lot of controversy about it. There are a report that shows it was only 30%. There are certain countries like Spain have reported issues with certain batch and reported only 30% sensitivity. So you have to be careful of what you're using as far as diagnostic tool. In this study, and you can maybe go back to this slide later and search this literature, they have shown the sensitivity of the lateral flow is 36.4. So when you look at the infection, the cycle of infection, so a patient that get infected will have the viral RNA for up to 28 days, and maybe a little bit more, but we're talking about a month where the RNA is still present in the patient, not at the same concentration. It peaks at 2 weeks and starts decreasing. The IgM, which is the first antibody, shows at the first week, the second week, at week 1, and then peaks at week 2 and decays around week 3. The IgG, which is the long-term antibody, it will start in 2 weeks, then it will peak at close to 3 to 4 weeks. So what does it tell you is there is an overlap between all these peaks, which means that theoretically, if you test RNA and antibody on the same patient, even if you have false negative rate, you should be able to get a low false negative rate, high sensitivity, if you are testing both RNA and antibody at the same time. This is how the antibody flow works. This is not the rapid test. This is the ELISA based test, a laboratory grade test that uses equipment. It takes 3 to 4 hours to complete. It uses a concept, similar concepts, which is the ELISA plate is coated with an antigen. When you throw in your serum, it has antibody. It will bind to this antigen. Then you have to wash the well, and then you have to add the conjugated antibody that is conjugated with an enzyme that will basically turn a blue substrate to a yellow color. That yellow color will absorb, so you have to have a plate reader and will turn that into a quantitative test, showing you the amount of antibody detected. In this study, this is the test that we currently use in the lab. So we have looked at, not us, but the manufacturer has done, in this case, 25 tests. They have looked at, all these tests were positive in PCR, and they were looking at the IgG presence. So they have seen that 2 out of 25 were negative in IgG. So we're talking here about 92% sensitivity. There was a study before this, a clinical trial, where they have used 30 confirmed positive and 54 confirmed negative, and they were all correlated. At Progenesis, what we've done, we've taken 5 patients that were reported to have been infected, and we looked at their IgG and in yellow, and then we took 5 negatives that we know they were not showing any infection, and they were negative in IgG, and we looked at positive control and negative control. So I think, I don't know how much time I have. I don't want to take too much time from this presentation, but basically, quickly, if you compare the rapid test and the ELISA test is in 92% sensitivity, while the rapid test is 30 to 36. And when you are screening your patients, for example, if that patient is negative by RT-PCR, you can retest. If they are negative by IgG or IgM, you can retest. You can also test simultaneously for IgG and RT-PCR. And this is the last slide, just to let you know that we have launched the first IgG test and the first RT-PCR test for IVF clinics. And we continue to kind of improve upon the test. And our really goal is to help both clinics and patients reduce the risk of infection and exposure. And with that, I think this is the end of the presentation. Thank you so much.